Welcome to the video on active transport. I hope the SATs went well if you took the SATs on Saturday. So we're going to begin. You should fill in the blanks, so you might want to pause the video, then fill in the blanks, and then listen to what I have to say. Remember, the goal is to annotate the slides or add things that I say to the slides. So let's begin. So far we've been learning about passive transport, and we learned about osmosis, the diffusion of water, facilitated diffusion, the diffusion of large and charged particles, and then osmosis. And then diffusion. So we learned about those three, and those are all forms of passive transport. When we were learning about passive transport, so passive transport was using no energy. It also moves things from high to low concentration. So active transport, keyword, the word active right here, is the opposite. It's using materials using energy, or ATP, which we're going to look at in the next couple of slides, and it moves things from low to high concentration. The key word here, or the key sort of word that's used to move things from low to high concentration, is to go against the concentration gradient. In passive transport, you're going down the concentration gradient. But in active transport, you're going against the concentration gradient or from low to high concentration. So the question is now, what type of materials need to go against their concentration gradient and from low to high concentration? There are some things that the cell needs in bulk. For example, one of them is glucose, and glucose is the main source of energy used for cells by the mitochondria in cellular respiration when they make energy. Cells need to get glucose into themselves. So cells need to get lots of glucose into their cells. And they can't just do it from using diffusion from high to low concentration. They need a lot of it, so it needs to go from low to high concentration. Active transport is actually also used by nerve cells to sort of allow them to transport information back and forth, which we're going to talk about much later around in March. But when there are some materials that cells need to get a lot of, or in bulk, and they use active transport, which is moving materials from low to high concentration, and it needs the use of energy, or the use of ATP. So active transport, just like facilitated diffusion, uses a protein. And there are lots of different types of transport proteins that are used in active transport. You do not need to memorize them, but there are some proteins that allow movement in only one direction. Those are called uniports. There are certain proteins that allow two things to move through the protein in the same direction. So we have like substance A and substance B. These are going to be used in something called co-transport, which we'll see in a second. And then we have an antiport, which is a protein that allows two things to move in opposite directions from each other. Kaiser is pressing the button on the computer. Stop, Kaiser. But essentially the point here is that active transport uses a protein. All forms of active transport, except for the last one that we're going to talk about, require the use of a protein in the membrane. So now we're going to focus on two different types of active transport. The first is called primary active transport, otherwise known as, and I have to write with the mouse, so this always comes out horrible, also known as an ion pump, which I'm trying to write, but an ion pump, which we're going to take a look at that first. And then the second one is called secondary active transport, which is known as co-transport. So transport, which takes me forever to write transport, but here would be the word transport. These are really, you should really refer to them as the ion pump and co-transport. We're going to look at the ion pump first, and then we're going to look at co-transport second. So primary active transport, or an ion pump, requires the direct use of energy in the form of ATP. So, ion pumps, one of the examples is the sodium-potassium pump, and the one that we're going to look at. And I apologize, Kaiser likes to write with the pen or step on the computer while I'm doing this because he wants attention, so that was what the delay was. So the most important thing you can know about ion pumps, or transport pumps in general, is that they use ATP. And ATP is this form of energy that the cell uses, so it can use ATP as a form of energy. All ion pumps use ATP to transport things, doesn't always have to be ions, but transport things against their concentration gradient, or from low to high concentration. Okay, we're going to now take a look at one example of an ion pump, which is the sodium-potassium pump. We're going to look at it in pretty vigorous detail here. So let's just get ourselves situated here. So here we have a membrane, and then inside the membrane is a protein. And if we take a look here, the first step, and you should annotate or say what I'm saying. I'm going to have trouble writing it down because I'm writing using a mouse. But what I say, you should write down. So you should probably pause the video often to stop and then write down what is said. So if we take a look here, the first thing that happens is that sodium goes inside the protein. And notice like it fits into the protein. Remember, these proteins are specific. And so this is the outside of the cell, and this is the inside of the cell. So there is high sodium concentration outside the cell, and we're trying to get more sodium outside the cell. So the first thing that happens is the sodium binds into the protein. And then this here is a molecule of ATP. If you take a look, what happens in the next image is that ATP binds to the protein and then loses a phosphate group. Okay, that loss of a phosphate group releases energy. So here we have a release of energy. This is called ATP hydrolysis. Okay, you're taking an ATP. Removing ATP means adenosine triphosphate. Okay, so you have an ATP molecule. Okay, and then you're breaking off one of the phosphate groups. These are unstable. We'll talk about this in the next unit while they're unstable. And then it releases a phosphate group. Okay, and that causes a release of energy. Okay, so here that energy is used, and notice that the protein changes shape or undergoes a conformational change. That energy, okay, is used to pump the sodium outside of the cell from low to high concentration. So the ATP breaks. Here the ATP is going on under hydrolysis to make ADP, and that energy is used to pump sodium outside the cell. Okay, notice that we still have this phosphate group here. Okay, what happens, okay, is that so the phosphate group is still here. Now the potassium, which is low outside the cell and high inside the cell, goes into the protein, and it has a binding site right here for the potassium. Okay, then this phosphate group leaves. This phosphate group leaves, causing a conformational change in the protein. Okay, and that conformational change, or sort of like induced fit changing, okay, causes the potassium to get released inside of the cell, and this cycle continues. But notice, okay, and this is sort of the key point of this. Notice that for every 3 sodium that get pumped out, this is important. So for every 3 sodium, so 3 Na, that get pumped out, okay, 2 sodium get pumped in. That's important. 2K get pumped in. Okay, this has a consequence. And if you take a look at this line represents our membrane. Okay. So because there are more positive charges, I mean, sodium and potassium are both positive. But because there are more positive charges being pumped out, the outside of the membrane, of any membrane, okay, so the outside of the membrane is more positive compared to the inside of the membrane. Okay, this sets up a charge gradient. There's more positive charges outside of the cell than there are inside of the cell. So it's like there's more positive charges on the outside than there is on the inside. This is important for how nerve cells function. The fact that there's a charge gradient across the membranes of cells set up by the sodium-potassium pump using the hydrolysis of ATP. Okay, but what's sort of the point? Do you have to remember the details of how, exactly how the sodium pump pumps the sodium out and the potassium in? No, but the important is it's using the energy of ATP by ATP losing a phosphate group to pump sodium out and potassium in. 3 sodium out, 2 potassium in, and it's using energy because the ions, or sodium and potassium, are going from low to high, or against their concentration gradient. So now we're going to talk about secondary active transport, or co-transport, which does not use and this one always confuses people, so I feel like you're going to be confused, and I'm going to try my best to unconfuse you, but this is always a source of confusion every year. So co-transport doesn't use ATP directly. What happens, in the picture will make more sense, is that it uses the energy of an ion gradient that's already been established. And we'll take a look at the image. The image will make it make much more sense. One of the examples that uses co-transport, which is an active transport mechanism, is glucose. So glucose is transported using this mechanism. All right. So this picture is important and sort of helps display how cotransport works. So here we have a proton pump, or an ion pump, which is using energy to pump hydrogen ions, okay, here's the ATP, against their concentration gradient outside of the cell. So here, the concentration of hydrogen ions is high. Like it's really, really high out here and really, really low inside the cell. So this, because of the laws of diffusion, okay, these hydrogen ions really want to rush back in. They really want to rush back into the cell. It's because they're really, really high outside the cell, okay, and very, very low inside of the cell because of this proton pump, which is continuously pumping hydrogen ions outside the cell using ATP. So since they're really, really high outside the cell, they want to rush back in. And they have this cotransport protein here that will allow them to rush back in. But here's where the cotransport part comes in. The hydrogen ions, they want to get in so badly, and they rush in through this sucrose cotransport here. And the energy of them rushing in back to the cell, because remember they're high out here and low out here, is used to pump sucrose against its concentration gradient inside the cell. So once again, I'll just say it one more time. So the hydrogen ions are high outside here because of the proton pump pumping them out, and they want to rush back in. Okay, and the energy of them rushing back in, going from high to low, is used to transport sucrose, which is a sugar, against its concentration gradient into the cell. So essentially, we're using the energy of a concentration gradient when the energy of this rushing back in is used to power something. In this case, it's sucrose against its concentration gradient. So now we're going to change gears to a separate form of transport. And there are some molecules that are just too big to fit through proteins. Like no matter if they're just too large, and going through a protein is not fit. Like the protein does not have a big enough opening for the particle to get through. And these particles use a process known as endocytosis, which is going in, or exocytosis, which is going out. And then instead of going through the membrane, what happens is that the membrane, this is the membrane, makes a pocket, okay, and encloses the particle. So here's the particle that's going in, and the membrane goes around it, and then it buds off in a vesicle. This process of going around it is called invagination, okay. And so the membrane goes around it, forms a small pocket around it, and then the pocket buds off, and when it buds off, it's called a vesicle, and the particle is then taken inside the cell. The important thing to realize here is that the particle is not going through the cell membrane or through a protein. The membrane is going around it and taking it in. On this slide, the only thing that's important to know, we don't need to talk about pinocytosis anymore. That word is no longer used. Another word for endocytosis is phagocytosis. There are white blood cells that function in your body, immune cells called phagocytes, and they eat bacteria and viruses that infect your body. But phagocytosis is just another word for endocytosis, or the membrane invaginating a particle and taking it into a vesicle and being inside. We are going to talk a little bit now about receptor-mediated endocytosis and how that's different than just regular endocytosis. Okay, so in receptor-mediated endocytosis, so these are the yellow particles, are the particles that we're taking in. The particles, instead of the cell just deciding to go around it, bind to receptor proteins in the cell, just kind of like transport proteins, except the particle doesn't go through. The binding of the things that want to get into the cell to the receptor proteins in the cell causes the membrane to start to invaginate. See here, the binding of these causes the membrane to start to invaginate. We don't care about the coated pit or the coated vesicle formation, then that signal. So them binding signals for the cell membrane to start to invaginate, and then it brings it in and forms a vesicle. So I guess to summarize, why receptor-mediated endocytosis is different than regular endocytosis is that it's more specific. There are specific proteins that cause the membrane to invaginate particles. You can actually block these receptors, and then the endo through competitive inhibition, I guess, and then the coat or the invagination won't occur, and this process will be blocked. We'll talk a little bit more about this in class tomorrow, but now it's time for you to go answer the guided questions or watch the video again. See you tomorrow.